Thank you. Good morning. I'll be talking with you about how we can get plants with deeper roots. And as you'll see when the images come up, this is the most important. Drought is the biggest limitation to plant growth on Earth. And when you have deeper roots, you do better under drought because the water can be found deeper in the soil. Of course, drought is going to be much worse in this century as a result of climate change. Another reason is nitrogen. This image shows regions in green where there's too much nitrogen, like in the US, where we're overloading the environment, causing pollution. In fact, nitrogen is the largest energy input in rich country agriculture, whereas in Africa, poor countries, they don't use nitrogen, and that becomes a limitation to food production. And the issue with nitrogen is, this is a simulation of what happens in May in Pennsylvania. That red is nitrate. Every time it rains, the nitrate is pushed down into the profile, and so the roots are trying to catch this resource before it is lost. So once again, deeper roots would help us get this resource. And the third reason is, this is an image from a beautiful study commissioned by ARPA-E, where they showed that deeper rooted plants would actually significantly improve carbon sequestration and build up organic matter in the US soils. In fact, their estimates are, under optimistic scenarios, up to 60% of transportation sector carbon emissions could be captured over 30 years just by having deeper rooted crops. So first I want to talk about idiotype breeding. It may not be a concept that's familiar to everybody. As opposed to brute force yield selection, why it's good to have an idiotype. Idiotype is an ideal type that you're breeding for. And this is, this is a fitness landscape, and if the environment is gradually changing, so the phenotype can have different optima. If you're just selecting for yield, you'll gradually track whatever optimum works for your environment. But real environments are not like this. Real environments are more complex. Even in one field, there's different environmental situations in different parts of the field, and so you have different fitness landscape in time and space. And there are many examples, such as in common bean, where they were at a fitness peak because of their breeding, and then they knew they had to get to a higher peak somewhere else, but to do that, they had to go through a fitness valley. They lost yield, but they ended up in a better place. And this is the reason we need idiotypes rather than just looking at yield, including genomic selection on yield. I'll be talking about this plant. This is the most important crop on Earth and in the US. And nitrogen and water are important for maize. Of course, in poor nations, as I mentioned, water and nitrogen are primary limitations to food security, where maize is a food security crop. Drought is the biggest risk to growing maize in rich nations. Nitrogen fertilizer is the primary energy, economic, and environmental cost of growing maize. The cost of nitrogen fertilizer has risen with energy costs. And amazingly, over half of the applied nitrogen is actually not taken up by the crops at all, but goes directly into the environment and causes pollution. And so this is a different visualization of what I showed a moment ago. The nitrate leaches down into the profile. The roots are growing, trying to catch that resource before it is lost. And so basically what we need to do is get as deep as we can, as fast as we can. So the idiotype we developed to help guide breeders in doing this is called steep, cheap, and deep. It's a combination of traits or phenes that help us get as deep as we can, as fast as we can to catch those resources. I'll be talking about a few of these today. First, steep. By steep, I mean architectural traits that position roots in different soil domains. Here's an example, optimal number of crown roots. What do I mean by that? If you dig up maize roots in the field, you see some, like on the left, have few crown roots. Those are the primary axial roots of the species. And the one on the right has many. In fact, if you look at 5,000 genotypes, you'll see variation from 5 to 40 crown roots per plant, significant natural genetic variation. This is a computer simulation of how this looks. So these first roots are seminal roots, and here come the crown roots. On the left, we have a low crown root phenotype. On the right, a high crown root phenotype. This is a structural functional model that allows us to look at the fitness impacts of these phenotypes. And this model told us that the one on the right is overkill. There's too many roots. They're competing with each other for internal resources, and they're competing with each other for nitrate in the soil. Plant doesn't need to do that. Contrary to maybe perhaps your intuition, the phenotype with fewer roots should do better, according to the model. And so you can see this in this visualization. As we zoom out, the one on the left, 46 nodal roots, it didn't get as deep as the one on the right because the one on the right had fewer roots. Each root had more resources, it could go deeper. To see if this actually worked in the field, we showed under drought that comparing lines that had few crown roots, many crown roots, that the lines with few crown roots had deeper rooting, better water capture, and between 60 and 116% more yield. For nitrogen in fields in the US and in Africa and in the greenhouse, once again, the lines with few crown roots had deeper rooting, better nitrogen capture, and 180% more yield. So that seems to be a good trait. Here's another one, few long laterals. So here's the phenotype we see in the field. On the left, we have the few long phenotype. On the right, the many short phenotype, the natural genetic variation. In silico, you can see what this looks like. So once again, those are the seminal roots. And now here come the crown roots. And on the right, you have the dense branching. On the left, the sparse branching. And the model tells us that, sort of like we saw with crown roots, fewer roots is actually better. The nitrogen capture is improved by having fewer roots fewer laterals that can grow longer into the soil and catch more nitrate. Once again, we have to confirm that in the field. So looking at natural variation for this trait, we see that the lines with few long laterals had deeper rooting, better nitrogen capture, better growth, and 32% more yield. Under drought, they had deeper rooting, better water capture, and 61% more yield. So by cheap, I mean traits that affect the metabolic cost of soil exploration, which in our analysis is important. And many of these are anatomical traits. I'm going to give you an example of this one, high RCA. RCA is root cortical parenchyma. This is not a simulation. This is an actual root. And that green space is airspace. This is formed naturally in all species, and it's stimulated by stress conditions. Here you see natural genetic variation. These two lines are actually sister lines of each other. They're recombinant inbred lines from the same parents. You can see on the left, intact cortex. On the right, some of that cortex has been destroyed by programmed cell death. And the idea is by destroying those cells, you significantly reduce the cost of maintaining that tissue, the carbon cost, nutrient cost of maintaining that tissue. This is what the phenotype looks like over time. So it's developmentally regulated. So the heat map shows increasing parenchyma formation. It's a complex expression pattern, and it affects respiration, nutrient content, nutrient transport, and other things, which are all built into this model. And so then the model predicts, for example, I don't know if you can see this well with the room lighting, but on the left we have a phenotype without parenchyma. On the right, an identical phenotype. That's something nice about computer modeling. You can make it identical except for the thing you're interested in, growing in a low phosphorus soil. And you can see over time the line with the parenchyma does better. And this is sort of a compounded autocatalytic effect where a small difference in the cost of growing means you can explore more soil, catch more phosphorus, more photosynthesis, and there's a feed-forward interaction so that over time there's a significant growth advantage from a relatively small difference in tissue cost. And once again, we don't trust these models. We just use them to guide our field work. So this picture is in Africa, but in Africa, in Pennsylvania, we see that these lines with more parenchyma have cheaper roots, deeper roots, and under nitrogen stress, 60% more yield. Under drought, we had a result with 800% more yield in the high parenchyma lines. In fact, we recently completed a study in Africa where we visited dozens of small farms in Malawi and looked at the lines they were growing. And actually, the farmers, as you can see here, helped us phenotype those lines. So we didn't intervene in the system. And then tracked the yield of those lines. And we saw that across those environments in Malawi, the lines that they were already growing that had high parenchyma yielded between 80 and 150% more in this naturally dry environment than lines without as much parenchyma. The large benefits we saw from parenchyma led us to think about other ways we could make roots cheaper. And so here you see natural variation for just the number of cortical cells. On the left, a line with fewer cortical cells. On the right, one with more cortical cells. Once again, from the cost-benefit perspective, it's better to have fewer cortical cells. And indeed, in these two locations in Malawi, you can see we had cheaper roots when we had less cortical cell files, cheaper roots, deeper roots, better water capture, and between 33 and 114% more yield. The blue bars are yield under well water conditions. There's no effect of these phenotypes I'm talking about when the plants are not stressed. They only show up under stress conditions. Here's another trait that people really hadn't thought about in plants. They think about this more in insects, and that's cell size. And cells are mostly vacuole, so by having a larger cell, you have multi-vacuole, which is much cheaper than cytoplasm. So we thought that having big cells would make a cheaper root. There's tenfold variation in cell size in maize. And we found that those large-celled lines had cheaper roots, deeper roots, better water capture, and between 100 and 150% more yield under drought. So there's been a couple of thousand papers written about water use efficiency, how shoots use water to gain carbon, but really what I'm talking about today is the other side of that equation, which is how roots use carbon to get water. And I think this is a useful perspective now to think about how to get better plants. Another challenge here, now this is a heat map of Arenchyma in maize at 42 days after planting. And now we're going to zoom into a functional structural model at a cellular and subcellular scale. And that these traits at the anatomical scale and the architectural scale interact with each other. You can see here, you probably can't see the text, but these graphs, this axis is Arenchyma, this is nodal root number. So there's an architectural trait I've told you about today, and there's an anatomical trait I've told you about today. Under low nitrogen, they interact. The line is not flat, it's curved. And if you increase the nitrogen, the interaction changes. And the same for phosphorus. And that makes sense. We all know that. Thumbs are great, but thumbs are especially great if you have fingers, especially if you're playing baseball. Not as much if you're swimming, right? So as biologists, we know that traits interact with each other in their fitness value and with the environment. But that's a problem for us. So what this means, so I've shown you today that in terms of maize yield under drought, there's a couple of architectural things I've mentioned and several anatomical things, and they interact with each other and with the environment to affect fitness. So identifying the number of traits we're interested in my team and the number of environments that are important, we've done the math, and there's actually 10 to the 23rd scenarios of interest. It's a large number. So we're using multi-objective genetic optimization algorithms to search through that space to find the best phenotypes to guide breeding. It's a complex problem. I just wanted to mention that the phenome is a very complex space. So phenotyping. I think in the introduction it was mentioned, something about tool development. So of course, looking at roots, there weren't good methods. So we've had to kind of make stuff up. And this is the first one, is shovelomics. Since this is a genome conference, we have to have an omics. You send graduate students out into the field with shovels. And actually, in 2 minutes per plot, you can get a lot of information. We're going through 20 or 30,000 plots a year in my program. With recent funding from ARPA-E, we're trying to develop a tractor omics, of robotics, which my students will be relieved when we develop that. But basically, you just dig up root crowns and wash them and then image them. And so Alex Bush has developed very nice software, DIRT, digital imaging of root traits, field portable, capture images, and then sometime later they can be analyzed when you have access to the internet. So for example, this is a variation for crown root angle in maize. Once again, in 5,000 lines, we see variation between virtually horizontal to virtually vertical. And doing GWAS after 4 years of shovelomics in Africa on these 500 lines of a diversity panel in maize, we see a hit from GWAS that's expressed in the right tissue, and a mutant in that gene has a steeper phenotype than the wild type in both greenhouse and field. And this is data from just Monday. Hannah shared this data with me. It turns out in field trials, those mutants actually have significantly more growth and nitrogen capture under nitrogen stress. So the shovelomics technique, as quick and dirty as it is, literally dirty, you get all muddy, actually is able to identify genes. For anatomical work, we started this with razor blades, and it was a very laborious process when you have tens of thousands of samples. So we came up with this laser ablation tomography system. And basically, you're using a laser to blast a tissue, and you're photographing it while you do that. And so this is a real-time image of a maize root being ablated by a UV laser. And then what you end up with is a bunch of smoke and an image that has all that information in it that then can be analyzed with image analysis software that we've developed. It can also be used in actual soil. You can see this is a UV laser, so the pink there, if you can see that, those are actually chitin, so fungi or insects. So then we can analyze these images in 2D, and we're going to be working on a 3D image analysis pipeline to pull out quantifications of these trait values. So here again as an example, here's for root cortical parenchyma, field phenotyping, shovelomics followed by laser ablation. So we have a hit from GWAS that's expressed in the right tissue, and a mutant has a highly altered parenchyma phenotype. So the challenge of translational research, and I'm not talking about it today, but we work directly with breeders in Africa and Latin America to try to move these into impact, is we're trying to have impact out here, and we're starting up here, and it's necessary, I think, to take a close look at the phenome. There are people, perhaps people in this room, who think you can go genes, yield, and that's it, and figure it out. I disagree. I think you really are served by thinking about the phenome. And if we think about the phenome, we have to think about phenes. Now, phene is a word that is 10 years younger than gene. 10 years younger than the word gene. It's the poor stepsister that never gets paid attention to. It is a word. I did not make it up. And the idea of phenes are they're elements of a phenotype. Just as genes are elements of a genotype, phenes are elements of a phenotype. Some fundamental thing that then they pile up and interact and form functions and performance and so on. Yield is not a phene. That's the key thing. In fact, genetics, excuse me, this is going to be a gross simplification. I don't mean to insult your intelligence, but just in a very simplistic way, genetics you could consider to be a process of discovering genes, understanding their function, and then genotyping to get more and more information, right? Like JGI. And most of the interest in phenotyping is driven by geneticists, because by phenotyping you can do gene discovery. However, I think this is giving the phenome and the phenes sort of short shrift, because there's also the function of these phenes. I'm showing you today original work on what's the value of large root cells for drought. That wasn't known. Or did people even think about cell size and breeding? No. So you could say that we discovered phenes, and we're trying to work out. So my service to breeders is discovering phenes, understanding their utility. I'm assuming you all or your colleagues are going to figure out the genetic basis, and then the breeders have a tool. And so that should be called phonetics. Once again, I did not make this word up. Here's a book from 1980. The Russians are way ahead of us. So in summary, we urgently need crops with reduced requirements for water and nutrients. The root phenome is the challenge and the opportunity to develop such crops. Idiotype breeding, guided by an understanding of the fitness landscape of specific phenes and their interactions with each other and the environment, is needed to develop these crops, in my opinion. As brute force yield selection, genomic selection on yield, I think will fail or will not get us where we need to be. Multi-scale mechanistic modeling is going to be indispensable because of the sheer complexity of these processes and interactions. The steep, cheap, and deep idiotype for water and nitrogen capture in maize appears promising. We have high-throughput phenotyping platforms, numerous genetic leads, so let's get busy. This is the team doing all the work. Thank you very much.
All right, I think we have time for a few questions. We've got some eager questioners right here. We'll get the mic to you.
Maize is mycorrhizal under natural conditions.
Yes.
And it can benefit from mycorrhiza-mediated mineral nutrient uptake.
Yes.
And help under drought conditions. Are you considering mycorrhizae at all in your modeling?
We do. And to a first approximation, we ignore them. Because they don't seem to explain anything important. All maize is mycorrhizal in all soils, so I mean, obviously it's cool and important, but working with breeders, we don't think it's something that is worth our attention. Maybe we should talk later about that.
All right, yeah.
So you almost touched on it a little bit, I think. But I'm curious, what are the biggest barriers that you've encountered to driving the adoption of the better yielding plants that is coming out of your research?
That's a big topic. We really don't have time to address it. The initial step is getting breeders to use these as selection criteria because they're very busy and stubborn, hard-headed people, and why should they look at roots? It's hard. And so there's a challenge in getting breeders to select on something that's different. And then we have developed, with our partners in Africa, Asia, Latin America, we've developed lines with much better yield, 2 or 3 times more yield in stressful soils. And they've been released. And then the challenge is, well, will people use them? In China, the government tells the farmers, you will use this. Huge impact. In Africa, where do they even find out about it? And it turns out the men want seeds they can sell in the market and drink more beer, and marry more women, because it's a polygamous culture where we work. And the women would prefer beans that don't sell in the market so they can feed their kids. So this is a really interesting social dynamic that we don't have time to get into, but it's non-trivial. That's a non-trivial issue, is how do you get this? Anybody, there's a lot of young people here, but if you have children, you'll know how this is. Like, I cannot get my son to eat broccoli. I've offered him money. Not going to do it. So how do you get people who are eating this color bean to eat this other color bean, even if it yields more? That's tricky.
All right, yeah, we got another one.
So how far can you go with cheap and deep before you encounter lodging problems?
We don't see any effect of these phenotypes on lodging.
All right, looks like we got another back here. Yeah.
Hi, very interesting talk.
Thank you.
So your focus was mostly on drought situations and what you're looking for. Is there any reason the phenotypes you identified would not be of equal value in, say, normal conditions?
Yeah, well, in the interest of time, we have a lot of work on phosphorus stress also. And it turns out there's a lot of trade-offs between traits for nitrogen and phosphorus. They're kind of very different resources and mobility and so on. And so this is one of the things plants are co-optimizing. Many cases, in fact, the ideal cases, as I've shown today, there's no effect of these phenotypes under non-stress conditions because something else like light or temperature is limiting growth. That's what we're looking for. But under stress, we see this differentiation. I don't know if that answers your question. So, under Iowa corn conditions where it's raining that year, it doesn't really matter what your roots are doing. You don't need roots very much. It's in Africa or in the future in Iowa where you're going to need these good root phenotypes.
All right, I think we can take one last question from Gloria here.
Hi. So I just wanted to know, so carbon, to make more roots, that's a carbon sink. And so I kind of had this feeling like making more roots sometimes isn't good for biomass. And so is there a sweet spot?
Well, I think you're absolutely right. In fact, our paradigm is not more bigger roots, but fewer, more efficient roots. Clearly. In fact, small is beautiful.
I support that.
Killing cortical cells, having less cortex. And I have stories that I don't have time to talk about. Root cortical senescence, fewer branches, fewer axes. It's like small is beautiful if they're good in a good place, right?
Okay, at 5'1" I agree with you. Small is beautiful. And I guess you're making those roots basically in the zone where nitrate is available in phosphate. In other words, where you make those roots. Like in Arabidopsis, that's a special zone too.
Yeah, exactly. We're optimizing spatial temporal localization of root foraging with resource availability, and that's how we're making progress. Thank you very much.
All right, thanks so much, Jonathan. Let's give him another hand.